जय हिंद दोस्तों। इस लेक्चर में देखेंगे रेप्लिकेशन ऑफ द डीएनए। वी विल सी दिस रेप्लिकेशन ऑफ द डीएनए इन दिस with these points: its meaning, then where it takes place, when it takes place, what is the origin, and the which enzymes are required for the replication of the DNA. First we will see the meaning. What is the replication of the DNA? Replication of the DNA, it is the process by which two new DNA strands are produced from the one DNA molecule. It is the meaning of the replication. Where it takes place? In case of prokaryotic cell, it takes place in the cytoplasm, while in case of the eukaryotic cell, it takes place in the nucleus. Then when it takes place, when the replication takes place, the replication of the DNA, it takes place in the S phase of the cell cycle. S phase of the cell cycle, G1, S, G2. This is the interphase, this is the S interphase, and in this interphase, during the S phase of the cell cycle, the replication of the DNA takes place. Then origin. In case of the prokaryotic cell, there is only one origin of the replication of the DNA, single origin of the replication of the DNA, while in case of the eukaryotic cell, there are multiple origins present for the replication, multiple origin of replication. So we will see the meaning. What is the replication of the DNA? Replication of the DNA, it is the process by which two new DNA strands are produced from the one DNA molecule. It is the meaning of the replication. Where it takes place? In case of prokaryotic cell, it takes place in the cytoplasm, while in case of the eukaryotic cell, it takes place in the nucleus. Then when it takes place, when the replication takes place, the replication of the DNA, it takes place in the S phase of the cell cycle, S phase of the cell cycle, G1, S, G2. This is the interphase, this is the S interphase, and in this interphase, during the S phase of the cell cycle, the replication of the DNA takes place. Then origin. In case of the prokaryotic cell, there is only one origin of the replication of the DNA, single origin of the replication of the DNA, while in case of the eukaryotic cell, there are multiple origins present for the replication, multiple origin of replication. Then the enzymes. Which are the enzymes that are? Then the enzymes. Which enzymes are required in the replication of the DNA, and what their function is? We will see the first enzyme. It is the helicase enzyme. What is the function of the helicase enzyme? You all know that the DNA, two strands of the DNA are held together with the hydrogen bond, and the breaking down of this hydrogen bond is the function of the helicase enzyme. अगर ये दो bond break हो जाते, then this DNA strand it will get separate. Means unzipping or the unwinding of the DNA strand is the function of the helicase enzyme. What is the function? Breakdown of hydrogen bond breakdown, and unzipping or unwinding of DNA strand. Then the next enzyme? What is the function of SSBP? What is SSBP? It is the single stranded DNA binding protein, single stranded binding protein or single stranded DNA binding protein. What is the function of the SSBP? Now this enzyme or the single stranded binding protein, it keeps the two separated strand away together, or it will prevent the two separated strand from further recoiling. It will prevent two separated strands from further recoiling. Then the topoisomerase enzyme. What is the function of the topoisomerase? What happens here, students? When the two strands of the DNA it get separated or it gets unzipped or unwound, then there is supercoiling will be produced in this region. Supercoiling will be produced in this region to release the tension that is produced in the supercoils. Yes, it is the function of the topoisomerase. So the topoisomerase it will cuts the DNA and reseals them, and it will decrease the tension that is produced in the supercoil. That is the function of the topoisomerase. It cuts the DNA and reseals them to avoid the tension that is produced in the supercoil. Then the function of the RNA primates. This RNA primates it helps in the synthesis of the RNA primer, and this RNA primer it will initiate the process of the replication. Then DNA polymerase. Actual function of the replication of the DNA, new DNA strand, it is synthesized by the DNA polymerase. RNA primer it initiates the process of replication, while DNA polymerase it continues the process of the replication. That is, the synthesis of the new strand of the DNA is the function of the DNA polymerase. And then the function of the DNA ligase. Now this DNA ligase it joins the Okazaki fragments. अभी Okazaki fragments क्या होते हैं वो देख लेंगे वीडियो में. Then we will see how the actual process of the replication takes place. Now the origin. We know in case of the prokaryotic cell there is only single origin for the replication of the DNA, while in case of the eukaryotic cell there are multiple origins for the replication of the DNA. What will happen? Now the DNA helicase it will break the hydrogen bond. It will break the hydrogen bond at the origin of the replication. Now DNA helicase it will break the hydrogen bond that is present between the adenine and the thymine nucleotide. Mainly the bond that is present between the adenine and the thymine nucleotide is broken because there is requirement of less energy as compared to break the bond between guanine and the cytosine nucleotide, in which there is triple hydrogen bond. Double hydrogen bond is easy to break. That's why the origin is at the adenine and the thymine base pairs. Then DNA helicase it breaks the hydrogen bond that is present between the two strands of the DNA, and the two strands of the DNA they will get separated. But because of polarity present between them, there are chances to recoil again. Then the enzyme single stranded DNA binding protein comes into play. It will attach to the two separated strand, and it will prevent the two strands of the DNA from further recoiling. As the two strands gets opened or they get separated, there is supercoiling over the strands of the DNA, and this supercoiling it is prevented with the help of the enzyme that is topoisomerase. Topoisomerase enzyme it will cut the DNA and reseals the DNA and resolves the tension that is produced in the stretch of the DNA. Then the actual synthesis of the replication takes place. Now the initiation of the replication of the DNA it is carried out with the help of RNA primer, which was synthesized by the enzyme that is RNA primase. RNA primase enzyme it will synthesize It will synthesize RNA primer, and this RNA primer is responsible for the initiation of the replication of the DNA. DNA polymerase is not able to initiate the process of replication. That's why this RNA primer, it will help to DNA polymerase to initiate for the replication of the DNA. RNA primer, it will bind to 3' end of the parent DNA strand, and it will produce the stretch of the DNA. Stretch of the DNA, it will be produced by the RNA primer. Means if there is thymine, its complementary nucleotide adenine will be bring out by the RNA primer. If there is cytosine, then there will be guanine. After that near about 10 base pair, after polymerizing 10 nucleotide base pairs, the DNA polymerase comes into play. Now this DNA polymerase enzyme, it will continue the process of the replication of the DNA. Now the direction of this new strand, it will be the 5' to the 3' direction. New strand is always synthesized. Remember, new strand is always synthesized in the 5' to 3' direction, or 5' to the 3' direction. Now look at this strand. The direction of this strand is 3' to 5', and the DNA is always synthesized in the 5' to 3' or 5' to 3' direction. Means DNA will be synthesized in this direction. The RNA primer, it will initiate the process of the replication of the DNA, and then DNA polymerase enzyme, it will continue the process of the replication of the DNA. The helicase enzyme, it will uncoil the two strands of the DNA, and the replication on the one strand, it will be continued in the 5' to 3' direction, while on the other strand there is a replication of this stretch of the DNA only, 5' to 3' direction. Look at this strand of the DNA. RNA primer initiate the process of the replication, it initiates the process of the synthesis of the new strand. DNA polymerase continues it. As the helicase enzyme moves, two strands of the DNA, they get separated. Now to start the process of the synthesis of the new strand, again another RNA primer will be required here, and there is synthesis of the new strand. That is in which direction? 5' to 3' direction. Again, as the two strands get separated, another RNA primer will be required. It will initiate the process of replication, and another strands of the DNA, it will be produced. On the other strands, there is continuously synthesis of the new strand towards the replication fork. But on the other strand, there is formation of the DNA fragments, and such fragments, these are called as the Okazaki fragments. These fragments are called as Okazaki fragments. Here, in one side, the synthesis of the new strand is taking place in 5' to 3' direction towards the replication fork, while on the other strand, the replication takes place in the 5' to 3' direction away from the replication fork. Now, these RNA primers, these are removed by the DNA polymerase enzyme. This DNA polymerase enzyme, it removes the RNA primer. And then comes the DNA ligase enzyme. This DNA ligase enzyme, it will attach, or it will ligate the Okazaki fragments. In this case, the DNA is synthesized continuously. That's why it is called as continuous or leading or forward strand. This is the continuous, leading, or the forward strand, while this strand, it is called as discontinuous strand, lagging strand, or the retrograde strand. This is the discontinuous, lagging strand, or the retrograde strand. Again, in the newly synthesized DNA, one of the strand of the DNA, it is from the parent, and other, it is the newly synthesized. While in another case also, one of the strand, it is from the parent, and this is the newly synthesized strand. In this case, one is from the parent, another is the newly synthesized. That's why the replication of the DNA, it is called as the semi-conservative nature of the DNA.